So if it's right there, then it's going to be
You
in the
need to
right
read
side.
class more
When you
entertaining.
put it in the left side like I did, you flip it.
Let's not do
It's
no,
like that.
let's just have a party.
Oh, then you do a
Yeah,
90
you
degree
don't
rotation.
your seventh graders get
Oh my
parties like
gosh,
every
this
week?
is way better. I don't have to do
Every
a
other
180. It's only a 90 degree.
Why?
All right.
This place is the best.
So let's just talk about sort of,
This year he has
guys,
parties for
Sylvia, our organization here. So finish biochem, then we started to move in and we talked about autotrophic nutrition, and we talked about photosynthesis, talked about respiration. Now where we're going to go is start to move through various organ systems, starting with heterotrophic nutrition, which would be what organ system? We're talking about complex animals. Yeah, so we're going to be really covering digestive system, and in simpler organisms, they don't have a digestive system. They have other adaptations that allow for the digestion of large molecules. So that's where we are.
So it's like me on Thanksgiving.
Does everyone know who that is? Ooh, good one. All right. So first of all, definitions. Heterotrophs. How could you define a heterotroph?
Eats things for food.
Okay, that would be one definition.
It doesn't make its food.
Doesn't make its own food. Okay, we're gonna step things up a little bit. And so what we're gonna say is a heterotroph is an organism that cannot synthesize organic molecules, basically food, from inorganic molecules. Autotrophs can do that. Heterotrophs need to acquire nutrients, acquire energy from consuming other organisms. So what are heterotrophs? What kinds of living things? Animals, all animals. Some bacteria. Anything else? What? Fungi. And we said animals, that would go for insects too. Some protists, yeah. So bacteria, the protozoans, if you remember we studied protists. Protists were broken into two categories, animal-like protists and plant-like protists. The animal-like protists are the protozoans. They're heterotrophs. Fungi are heterotrophs, animals are heterotrophs. So all of these organisms need to acquire organic molecules to use for energy, and then usually break them down so that they can be utilized within that organism. And when we talk about how living things get their nutrients, how they obtain nutrition, we can split it into sort of three main steps. Ingestion, digestion, and egestion. Ingestion is what? Taking in the food. Digestion.
You're ingesting your
I am ingesting some nutrients right now.
That's not an apple, it's a pear.
What else? So what is the definition of digestion?
The brain.
Breaking large molecules into smaller molecules.
Like the Krebs cycle for glucose.
Okay?
Hey.
Let's say glucose being digested, broken down. Technically that would be correct. And when we talk about digestion, we often talk about mechanical digestion versus chemical digestion. So as I take a bite of this pear, and chew it up,
You guys were listening.
I'm mechanically digesting it. What is the purpose of me chewing up this pear? Why don't I just swallow this chunk of pear?
Because it You're
gonna
stuck
get
in
puzzled.
your throat.
So I can probably swallow it, it might hurt, but
Increasing surface area.
physically breaks it down, increasing the surface area on which enzymes can work. Digestive enzymes can start to break it down chemically. That's why we chew. It increases the surface area of the foods we're ingesting. So how is chemical digestion different?
Enzymes.
It involves enzymes. Saliva is an example.
So,
Yeah, so when I chew up this pear and swallow it, it's still pear. But when foods are chemically digested, yeah, they're actually changed into other types of molecules, other compounds. And chemical digestion works through a process that we call enzymatic hydrolysis, which you know what those words mean. Basically, enzymes are involved in the hydrolysis of large organic molecules into smaller molecules.
Can you explain the picture to me? Like I don't understand what's supposed to be like funny about it. It just
Heterotrophs win.
You could
yeah, but
probably.
like I don't get it.
It's okay. Don't worry, man. I don't think it's really that funny, but it was appropriate. I was searching for heterotrophs, I found it. And finally, often after digestion of material, there's a need for egestion, which is the removal, the elimination of anything that was undigested. Not all molecules can be digested by various organisms. Sometimes they don't lack the proper enzymes to digest certain types of molecules, and therefore there's undigested material that needs to be eliminated from the organism. So we think back to hydrolysis in the last unit. Hydrolysis is splitting a large molecule into smaller, often more soluble molecules. Things that can be absorbed into the cells of a living organism. And we know that there are many different types of enzymes that allow various types of molecules to be hydrolyzed. You know, an example, starch being broken up into smaller glucose molecules. That's hydrolysis. Or disaccharides, polysaccharides. Water is added, and they get broken down into smaller subunits, monosaccharides, which can be absorbed by the organism. So there's lots of types of enzymes that do this. We know that they end generally in the ASE suffix, lactase, sucrase, amylase. What does lactase break down?
Lactose.
The sugar lactose. Sucrase, sucrose, table sugar. Amylase, starch. Yeah, that breaks down starch. Proteins are broken down by hydrolysis into their component amino acids. Do you know the name of the enzyme that would do that? Protease, yes. There's different types of proteases. What about lipids? Let's finish up this slide first. What's that? Lipase breaks lipid into its components, which are fatty acids and glycerol. So their enzymes are specific to certain large molecules, and they only will act on those molecules. Okay, when you guys are done with that, you can take a break till the bell rings.
That's the same thing as like
Is it what?
Not really.
All right, so the what we're going to be doing now, as we move forward throughout these units, we're going to be talking about different adaptations that different groups of living things have that allow them to go through certain processes. Like, for example, we're talking about nutrition and digestion. So we're going to talk about, okay, well, how does that happen in bacteria? How does that happen in protists and fungi? And then we're going to talk about some representative animals, right? Where the earthworm, the grasshopper. And we'll talk about them for each of these different body systems. So let's start with bacteria. So bacteria are heterotrophs, mostly. So they also, though, they're single cell. We know that they're prokaryotes. And so for them to absorb materials, ingest them, basically they ingest material through diffusion. And they can break down molecules, but they do it using a method that's called extracellular digestion. So we're going to talk about in these organisms extracellular digestion versus, you know what the opposite would be? Intracellular digestion. So extracellular digestion means what?
Out of the cell.
Yeah, it happens outside of the cell. The digestion takes place outside of the cell. Intracellular means happens within the cell. So bacteria have extracellular digestion. They secrete digestive enzymes, release them onto the material that they need to digest. And once it's digested outside of the cell, it can then diffuse into the cell. But the actual breakdown happens outside of the bacterial cell. And bacteria, kingdom Monera, is the older name for the kingdom of bacteria, but are very, very important for human health as well. In our digestive system, we have a whole host, a whole ecosystem of various types of bacteria that help us in our digestion of foods. Okay? They're called gut flora. Okay? Living things inside of our digestive tract that help us to digest our foods. In fact, I just printed off an article for you guys to read later on in the unit about means of treating certain digestive diseases by giving a person a transplant of good bacteria into their digestive tract. We'll talk more about that when we get to humans. Fungi. So we know fungi are heterotrophs. Mushrooms, mold, yeast. Okay, they're not autotrophic. And fungi are often saprophytes. They live on dead, decaying organisms. Okay, if you imagine some of these things, this, moldy strawberries, or these mushrooms growing out of some cow poop, or mold on these leaves, or mold on this bread. These are all fungi, and what they do is they secrete, they have these things that are kind of like roots, although they're not roots, they're called hyphae, hyphae, and they actually produce enzymes, release them onto the dead, decaying organisms that they're breaking down. They break down that material, and then the fungi can absorb through those hyphae the nutrients that have been produced. So again, that's extracellular digestion. The enzymes are being secreted, they leave the cell, they break down molecules, and then the nutrients are absorbed. Protists. We studied protists, we studied the amoeba, we studied the paramecium. How does the amoeba obtain its food? Through pseudopods, through engulfing its food particles. Okay? We call that phagocytosis. Surrounding a particle of food, engulfing it, and once it's engulfed, where is that food particle? After it's been engulfed. It's inside of the amoeba. It's in a food vacuole. That is where it gets digested. Amoeba are single-celled, but they have lysosomes. Small membrane-bound sacs that have digestive enzymes in them. When these lysosomes merge with a food vacuole, they release those enzymes, which can then break down the food particle. And then that digested food can diffuse into the cytoplasm. So what type of digestion is this? Yeah, this is an example of intracellular digestion. So here we have an amoeba engulfing a bacteria. Surrounds it, the pseudopods engulf it through phagocytosis. It's then inside of a food vacuole. The lysosome comes over, emerge, and then those molecules of food that were ingested get broken down. Any remaining undigested materials egested through exocytosis. Okay?
Are we going to go over cytosis?
What's that? No, not in this one. Maybe on the SAT too. Yeah, it's just a matter, it's really just a variation. Really. They're pinocytosis, phagocytosis, just a variation of each other. Okay, where it's not an engulfing, but sort of pushing inward rather than going outward. Okay, paramecium, a ciliate. They have that pellicle surrounding them, that sort of hard outer shell, so they can't engulf their food like an amoeba would. Because they don't have that flexible membrane on the outside. So instead they have an organ that they use for ingesting food called the oral groove. The oral groove has cilia in it. They kind of guide in particles of food. They go down the gullet of the paramecium into a food vacuole at the end. And then digestion takes place just like it does in the amoeba. Lysosomes merge, break down the food. And again, egestion happens through a special organ in the paramecium called the anal pore. Because there's that pellicle, that's the way for material to leave the paramecium. So again, your food molecules get swept into the oral groove. They travel down the gullet into a food vacuole, okay, where it then gets digested, and then any waste comes out through that anal pore.
You still ripping that book?
Why are you ripping up the book?
All right.
What book is that?
All right, so let's hold it until later. How about? Okay.
Wait, why is he ripping it?
Don't worry about it. So now, as we move from bacteria into protists, and now then fungi, now back into the animal kingdom. So we'll talk about some of those representative animals. Animals that we study to see how sort of more complex animals go through these life processes. So the hydra. Okay, we've seen hydra. In 7th grade. Hydra is a cnidarian, also called a selenorate. They mean the same thing, they're just different names for the phylum. Okay? Hydra are a pretty simple animal. They have sort of a digestive cavity, okay, just sort of a hollow opening inside. One body opening, okay, for food molecules to come in and waste to leave. And then they have obviously those tentacles. Okay? The tentacles of the Hydra are used to ingest their food. Okay? So the Hydra has on those tentacles these stinging cells. Nematocysts is the name for them. You can look at what they look like here. The tentacles, so this is the whole tentacle, and inside this is the nematocyst. It has sort of a coiled up little harpoon sort of thing. When it discharges, okay, it uncoils, sending the barb out. It's attached here to a thread, and it sticks into the organism it's preying upon, and immobilizes it. Okay, and then they can eventually use the tentacle to put that their prey into their body opening. Again, if you've ever been stung by a jellyfish, the reason it hurts is because these barbs from these nematocysts of a jellyfish are sticking into your skin. Okay, and it burns. So those nematocysts help to mobilize the prey, and then they put the prey into their mouth, into their digestive cavity, this hollow space that's in between them. And then they actually can perform digestion two ways. They have extracellular digestion and intracellular digestion. Okay? Extracellular digestion, I'll let you write that down first. So the Hydra has just two what we call germ layers, like its tissue of its body have just two layers. Okay, there's the endoderm, okay, which is on the inside, and then the ectoderm, which is on the outside. So like those nematocysts, they're on the ectoderm. But the endoderm is the internal cavity of this Hydra, the cells facing the inside. Okay, and what happens is those cells in that endoderm, they can release their enzymes into this body cavity. So after the Hydra ingests its prey, okay, then these cells on the endoderm, they can release these digestive enzymes. And that starts extracellular digestion. Those cells in the endoderm, they have flagellum to sort of mix around the food, to move it around, circulate it so it can be digested. And they also have cells on that endoderm that can engulf these food particles as they're being broken down and bring them into the cells of the endoderm. Once they're inside of the cell, intracellular digestion can occur through processes similar to the amoeba or the paramecium, where you have the lysosome breaking down those particles using digestive enzymes. All right, any questions up to this point? We're going to actually stop here in our notes for today.